You're muted, Stephanie.
Thank you. Sorry about that, everyone. Good afternoon. I'll try that again. Welcome to Ocean Expert Exchange. Scientists in every Florida school and the ANGARI Foundation are excited to have you join us for another live webinar event on ocean science and technology experts. In this monthly series, we dive into all things marine science and explore what's happening in the field, interesting careers, and more. So today we'll be speaking with Dr. Dean Grubbs of the Coastal and Marine Laboratory at Florida State University about his research on small-toothed sawfish. But before we turn things over to him, we'd like to share just a little bit about our programs. The Scientist in Every Florida School is a free program housed within the Thompson Earth Systems Institute at the University of Florida. The mission of CEFS is to engage Florida K-12 students and teachers in cutting-edge research by providing science role models and experiences like today that we hope will inspire future stewards of our planet. The ANGARI Foundation is a nonprofit headquartered in West Palm Beach, Florida, and the foundation supports and promotes marine science research and education, and many of the foundation's primary initiatives involve its 65-foot research vessel, the RV ANGARI. And in case you missed any of the information in today's preview slides, we'd like to remind you that we'll be taking any questions you submit for the scientist at the end of the presentation, and you can put those in the chat box at this time and throughout. So at this time, I'm going to stop share and turn things over to Dr. Dean Grubbs. Dr. Grubbs, go ahead and take it over from here.
All right. Thank you for the introduction. Hopefully you all can see my slides now. So thanks for the invitation. Thank you all for coming. Good afternoon, good morning, evening, depending on where you all are. So my name is Dean Grubbs, and I'm the Associate Director of Research and also a research professor at the Florida State University Coastal and Marine Laboratory. In my lab, most of our research involves studies that address specific questions needed for either fisheries management or for conservation of marine fish stocks. Most of the work that we do is on sharks and rays, but we also do work on bony fish and other things. We do a lot of work looking at what drives fish communities, what drives their food webs. We look at life history patterns. How long does it take them to reach maturity? What are the reproductive patterns? Those kinds of things. And we also look at ways to mitigate for things like bycatch, mortality, oil spill effects, even climate change. And a lot of the work we do involves looking at habitat use and migration patterns using every tool of tagging and telemetry under the book. And first, I'd be remiss without first acknowledging all of my graduate students. So all the work that I do in my lab, it's really the graduate students that do the bulk of the work. And so just shows you some of my current graduate students and some of the former graduate students in my lab who are now all gainfully employed across the spectrum from federal agencies to universities and everything in between. Now, the work that we do, we work in a number of different places around, primarily in Florida and in the Gulf of Mexico. And so we do work that is basically looking at the biology and ecology of economically important or poorly studied fishes. And so we do work on deep sea ecology. You see my pointer's not showing up. There we go. Deep sea ecology, some in the Bahamas, but most of that work is in the Gulf of Mexico, and it's work that's in response to the Deepwater Horizon oil spill. Get my screen back up here. And we also do a lot of work on coastal shark and bony fish communities. This is mostly distributed in the Big Bend of Florida as well as the Panhandle of Florida. This little star shows you where the FSU Coastal and Marine Laboratory lies. And then we do work on small-tooth sawfish, which is the species I'm going to talk to you about today. Most of that work on the small-tooth sawfish is in the state of Florida. And you're going to learn all about what a small-tooth sawfish is here in a second. But we also do work in the Bahamas on small-tooth sawfish and a little bit of work in Cuba even. And so that's the species I want to talk to you about today. And so we've been studying small-tooth sawfish now for about 17 years since I got to FSU, and it involves a lot of collaborators from a lot of different agencies, as well as a lot of my graduate students that are doing this work. And so I'll just tell you in real brief detail some of the lines of research we've done to promote recovery of small-tooth sawfish and why we need to do that work. And so I'll start by telling you a little bit about what a sawfish is. So sawfish are a small family of rays. There are 5 species in the world. And so not a small family of small rays. They're all very large. 4 of the 5 species get at least 17 feet long. So they get huge. We have one species in the United States, and that is the small-tooth sawfish, Priscus pectinata. Now, when I say they are rays, that means they're more closely related to the stingrays than they are to sharks, even though superficially they look sort of shark-like. But if you look at this is the living diversity of sharks and rays and their relatives in the world now. There are 14 living orders. You've got 9 orders of sharks over here, and then you've got 4 orders of batoids, those are the rays, and then there's chimeras here. So the sawfish are over here as batoids, even though they look superficially like a shark. So what's the difference between a shark and a ray? We like to affectionately call the rays pancake sharks because they're usually just sort of flattened. But the big difference is if you take the pectoral fins and fuse them to the head so the gills are underneath, then you're a ray. If those gills, pectoral fins are not fused to the head, then the gills are on the side, and then you're a shark. Also, the rays have these fused vertebrae in right behind the skull that basically reinforces their skeleton to allow them to flap those big pectoral fins. The sharks don't have those fused vertebrae. This is a lemon shark right here. And so, if we look at a soft shark, you'll see that it's got its gills on the side. These are relatively small deep sea sharks, whereas the soft fish, their gills, you see, are underneath on the bottom, and they are large, shallow water tropical rays. Now, it may surprise you that small-tooth sawfish is a top predator in its system. You hear that term all the time, apex predator or top predator. Most sharks are not top predators. Most shark species are right in the middle of the food chain. But some sharks, like great hammerheads or white sharks or bull sharks, are at the top of the food chain. And this is a sawfish here with an adult Atlantic sharp-nosed shark hanging out of its mouth. This is a black-nosed shark that's been hit on the side by the rostral teeth of a sawfish. And so that's the key character, right? These long is elongated rostrum with these teeth on the side. That adaptation's actually evolved in 4 different groups of sharks and rays through evolutionary history, but now we only have the 5 sawfish and 10 saw sharks that have that solid rostrum. So these guys are major predators in the systems where they live. In fact, this is an analysis we did. You don't need to get into the details on this, but basically, the higher you are on this graph, the higher you are on the food chain. This is called a stable isotope analysis. The black dots are sawfish, the white dots are bull sharks, and you see that they overlap. What that tells you is that sawfish are basically in the same at the same food level, the same trophic level as a top predator as bull sharks, all the way from the time they're both babies till the time they're adults. Now, what's the problem? The problem is that all 5 species of sawfish in the world are critically endangered. The species that we have is the small-tooth sawfish. It historically was on both sides of the Atlantic Ocean. Now its distribution is primarily in Florida and adjacent regions, with a few sawfish remnant populations in Central America. Why are they endangered? Well, historically targeted commercial fishing. This is a fishery in the Florida Keys about 100 years ago, targeting sawfish. They're coastal and easy to target with that big toothed rostrum. Sport fishing. It became vogue in the middle of the 20th century to go down to the Florida Keys and just catch a big sawfish, kill it, and take the rostrum off of it and hang it on your wall. Bycatch is a huge issue. With that big, long-toothed rostrum, they get caught in any sort of entanglement gear, such as a gill net or a shrimp trawl. And then finally, habitat loss. Sawfish as juveniles are obligately tied to red mangroves, which you can see right here. And we, of course, lost a lot of our red mangrove habitat. Mangroves have been declining globally, and we've lost a lot in our region as well. U.S. and Florida was relatively proactive in protecting sawfish, which is probably why we still have sawfish today. They've been protected by the state of Florida since 1992. Gill nets were banned in Florida waters in 1995, which probably did a lot to protect juvenile sawfish, particularly down in Southwest Florida. In 1996, the species was listed as critically endangered by the International Union for the Conservation of Nature that produces the Red List. In 2003, they became the first marine fish, the first native marine fish ever listed as endangered under the U.S. Endangered Species Act. So they've been listed as endangered for just over 20 years. And in 2007, they were listed under Appendix 1 of CITES. CITES regulates international trade for all countries that sign on to that treaty. That means since 2007, it's illegal to sell any product from a sawfish between countries. That doesn't necessarily mean you can't catch them within the country, but you can't sell them between countries legally. Unfortunately, there's still no protections for sawfish specifically in the Bahamas, an adjacent population. That'll become important here in a second. And so the work that we've been doing over the years is looking at the distinction between the U.S. and other populations, like the Bahamas. We're looking at the reproductive biology, trying to figure out what their population is doing over space and time. Are they recovering since the endangered species listing? And what isn't the critical habitat that a sawfish needs to survive? And then finally, we're trying to figure out ways to decrease bycatch in different commercial fisheries. So we're asking questions like what characterizes critical habitat from the time they're born until the time a sawfish is an adult? Where's the highest risk that a sawfish might interact with a fishery and get caught and die? Is the U.S. population its own population, or is there movement between Florida and other populations? And if so, then where are the other places outside of Florida that sawfish are giving birth? At the time we started this work, we only knew sawfish were giving birth in Florida. Now, how do we do this work? This kind of shows you, this is from a Changing Seas documentary we did a few years that shows you how we catch our sawfish. We basically set, and we'll be going down to the Keys doing this next week. We set a scientific long line. It's 50 hooks set for exactly 1 hour. We do it 1 hour because we don't want to kill anything that's caught on board, that's caught on it, like sharks. Once the sawfish is captured, we have to get it under control. These animals are incredibly strong. This one you'll see here almost ended my career when it almost hit me in the head. But they will knock a hole in your boat. They're very strong animals, and so we get them under control and then measure them. We take all of our samples, such as genetic samples, blood for stress physiology and reproductive analysis, muscle to look at their where they fit in the food chain, and then we tag them with whatever tag we're going to put on them, and will then release the animal. This shows you where we've been doing our work over the last dozen years or so. This is actually a sawfish here that we caught off Cedar Key, which is the furthest north that we've caught an adult sawfish. We caught this one summer before last, and you can see there how strong they are when that animal tried to come in the boat. But we've done over 1,000 sets. And these little green dots show you where we've done a set to try to catch a sawfish, and the yellow bubbles show you where we've actually caught them. And the places that we really catch, and you can see here that we've caught 122 large sawfish on these lines, but over 4,000 sharks. So we catch a lot more sharks than we actually catch the endangered species we're going after, which makes sense. The areas that we catch them. are primarily in two regions. One is in the northern part of Florida Bay and Everglades National Park in very murky creeks and bays that look like chocolate milk. This is primarily in the spring and summer, and it's mostly adult males in this region. And then on the edge of the continental shelf in the Florida Keys, from all the way from the middle to upper to the lower Florida Keys, in areas where it's more like 150 to 200 feet deep. And we get both sexes, males and females, together on the edge of the continental shelf there. Now, among the tools we use to study these animals is implanting acoustic transmitters. We've also done a lot of satellite tracking of these animals. But if we implant a transmitter like I'm doing right here into the body cavity and we put a little suture in it and sew them shut, that transmitter will emit a little acoustic ping every couple of minutes for 10 years. And there are receivers distributed all through Florida and beyond in both the Gulf and the Atlantic sides, and in all the other states as well. And if one of these sawfish passes by any of those receivers, then it gets picked up. And so this allows us to understand some behaviors that we would have never been able to get to without this technology. I'll just show you one quick example here. This is an animal we tagged down here in the Florida Keys, 1st of April 2017. 3 weeks later, it was all the way up into Cape Canaveral. At the time, we didn't even know sawfish used Cape Canaveral very much. The animal went back by May down to the lower Florida Keys, then went all the way up to Tampa, offshore of Tampa, back to the Florida Keys, messed around in the Everglades over the winter and spring, and then showed up back in Cape Canaveral the exact same day, the 23rd of April, the following year. Then it did the same kind of thing, showed back up there the 19th of April. And so this animal actually for several years in a row showed up in the same spot in Cape Canaveral each spring. We would have never gotten that kind of information without this tagging technology. And so this shows you sort of where we've tagged all of our animals down in South Florida, most of them at least, but we're able to get detections of animals far away from this area. We've actually picked up detected sawfish we tagged in the Florida Keys all the way up on our own receivers in the Florida Panhandle, and they've been detected as far north as Charleston, South Carolina. And so these using this telemetry, both satellite and acoustic telemetry from juveniles and adults, we've been able to answer a lot of questions about the movements and migrations and habitat use patterns of sawfish. This is just a few of the papers that we've published from this. I'm not going to go into detail here. I'm happy to share them with anybody who wants them. One of the big questions that we had is this animal here sort of illustrates it. This is a large male, about 13 feet long, that we tagged in the lower Keys. That after we tagged it, it went up to Cape Canaveral like that other one, and then it went all the way up to Georgia and got caught in a shrimp trawl. And so animals caught in shrimp trawls are probably going to die. And so we started looking at ways to figure out where sawfish are most susceptible to getting caught in shrimp trawls. And so we use both this as acoustic telemetry detection, so where sawfish are picked up on those receivers. These are satellite tracks of sawfish. And my former student Jasmine Graham combined these along with data on the distribution of gillnet fisheries in green, longline fisheries in orange, and shrimp trawls in magenta. And we figured out where is the peak overlap between sawfish and these fisheries. And we use that essentially to try to suggest to the federal government places where we might want to have closures to shrimp trawling. Now, what about this question of other populations? So we found out pretty early that in the Bahamas, there are sawfish, and they're mostly on the Great Bahama Bank between Bimini and Andros. And so over a number of years, 10 years or so, we did 15 expeditions to Andros to tag sawfish. And after doing a lot of satellite telemetry and acoustic telemetry, we found that there seems to be no exchange. The ones tagged in Florida never went to the Bahamas. The ones tagged in the Bahamas never went to Florida. And so that led us to this question of, well, if they're separate populations, then where are they pupping in the Bahamas? And this leads me to one of my favorite things that ever happened, which was in 2016. We were in the Bahamas. This is the first time this was ever observed in the wild. We caught this large adult sawfish here. It was the first big adult we'd ever caught, and in the Bahamas, that is. And you'll see we're going to get her tied up along the side of the boat here and get a rope on her, get her under control. You're going to see that my student will jump in the water with her pretty quickly to take blood samples. We take those blood samples to look at reproductive hormones. That tells us whether she's pregnant or mating, and then also that gives us an idea of how stressed she is physiologically. And so Bianca, my former student, gets blood to do those samples. But when we turned her over to tag her, we noticed movement in the cloaca right here. And this is the first time this had ever been observed. That is two sawfish rostra, baby sawfish rostra, that are trying to get out of the cloaca, trying to be born at the same time. They both couldn't fit out of this cloaca, so I got to basically deliver sawfish pups. It's the first time this had ever been seen. Um, and there were three that were trying to get out. We delivered those three. We didn't mess with any that weren't trying to be born. You'll notice there's no yolk sac. They basically use a yolk sac until they're ready to be born. The fact that the yolk sac is gone tells us that these guys were ready to be born. In fact, they were bigger than some of the ones we catch free swimming in the Everglades. Um, Notice there's this sheath, this protein sheath around the teeth that protects the mother as well as protecting the protecting the pups. We put a little microchip in the babies, like you would put into your dog or your cat, and then after that we release them. Now, while we were getting ready to tag her, you'll notice that the mom knocked my colleague, Dr. Andrea Krutz, backwards there. When she did that, she birthed two more sawfish babies on her own. And so we worked up and took samples from these five baby sawfish. One of the two that she birthed disappeared in the murk, but you'll see it come swimming up here in a second, and then they basically will catch it and tag it, and then once we're done working these animals up, we essentially release the animals. And so you'll see here the first footage ever of a newborn sawfish in the wild swimming off after being born. And so this is one of the coolest experiences that I've ever had for sure in my days doing sawfish research. Now, one of the cool things is that the reason we probably still have sawfish in Florida is that a lot of their habitats protected is national park like the Everglades, national wildlife refuges. The same is true in the Bahamas. West Side National Park and Andros protect some of their habitat. And so having protected habitat is probably critical to having these lifeboats for recovery. Now, one of the things we noticed is that when we compared our attempts to catch sawfish in Florida versus comparing them in the Bahamas, we caught way more sawfish in Florida than in the Bahamas, about 10 times more. And so this shows you our catch rates of sharks in the two different regions in Florida and the Bahamas, and they're about the same, but our catch rates of sawfish are way higher in Florida. So that tells us this the recovery of sawfish may really rely on the state of Florida because this population is by far the largest population. Now, we also found that there's a second smaller population of sawfish in the Bahamas around Abaco, in Little Bahama Bay. And we have a paper that's coming out any day now that shows that there's also a live population in Cuba. But most of the largest population by far is in Florida. And so recovery of the population probably relies on the United States. And so I started this all mentioning that our work is really research to promote recovery. Well, what about this question of recovery of the population? Is there any hope for them? Well, the sawfish have been listed over 20 years on the Endangered Species Act, and the whole goal of the Endangered Species Act is to get the animal off the Endangered Species Act, to be able to delist them because they recovered. And we were actually seeing signs of a positive recovery. This is an index of abundance showing the population recovering. And the fact that we're catching sawfish further and further away from their core habitat tells us the population is expanding. Unfortunately, you probably heard that last year, this year actually, starting last winter, there was a mortality event for sawfish in the Florida Keys, and we know of at least 56 adults or large sawfish that died. And so we think we figured out who the culprit was for that, what was killing them. And our work this year will really revolve on figuring out how big of an insult was this to recovery? How much did this set us back in recovery? And with that, I'm going to stop and say thanks for listening. These are two of the books that a colleague and I have written on shark biology. One is sort of a textbook for anybody who wants to know about shark biology. The other one's more of a natural history of sharks if you're interested. So that's a shameless plug for those. And then I'm happy to answer any questions you may have.
That was fantastic. Thank you so much for sharing about your work with us. I really enjoyed learning about the types of things that you do. We're going to go ahead and transition, as you mentioned, to our question and answer session for today's presentation. And we're going to go ahead and start with Angie, who says, you see so few animals with rostrums. Why would a sawfish evolve to have one?
That's a great question. And I would like Angie to tell me the answer. So it's interesting that in the sharks and rays, you have four different groups that have independently evolved this sawed rostrum, but they're all different. Each time it happens, it's different. Like even with today's saw sharks and saw fish, they're different. The saw fish has these teeth that are about the same size and are blade-like, and kind of sit in a socket in there, whereas the saw sharks have these narrow conical teeth that vary in size and are attached to the rostrum more firmly. Now, generally, they're used for, in sawfish, that this rostrum is used for feeding. It's used for basically protecting themselves from predators, but also disabling their prey. And so I showed that picture of the black-nosed shark with the holes in its side from being hit by a sawfish. We regularly catch sawfish with big scales on their sides, and to that point, I see somebody asking about the prey. They're all fish eaters. The sawfish are fish eaters. They're eating only things like we find the scales of red drum, redfish. They eat things like bonefish and snapper and ladyfish. They love ladyfish. They'll eat small stingrays, obviously small sharks. And so they use that rostrum to immobilize their prey. But it's always, every time that sawed rostrum has evolved, it's a small group. Five sawfish, 10 species of saw sharks. And you don't really see any intermediate in the fossil record, any, sawfish with real short rostra. It's like this was a relatively quick, um, evolutionary adaptation, a mutation, basically. Um, which is kind of similar to what our colleague at the University of Florida, Gavin Naylor, has found with hammerhead sharks, that, you look at the size of the hammerheads and how big their heads are, with like the bonnethead shark we have having a really small head, and then you've got the great hammerhead and the winghead with really big heads. And you would think, well, maybe the ones with the big heads are the most ancient ones, are the most recently derived ones, rather, and the small heads are most ancient, but it's the other way around. The big headed ones are the ones that are the most ancient. So they all of a sudden, in evolutionary timescales, all of a sudden develop this mutation of this big head, and it's getting smaller. And so I don't know the answer to why there are so few species that have this solid rostrum. The sawfish are part of a bigger group that includes guitarfish and wedgefish that do have a calcified rostrum, but it's not overly elongate and doesn't have teeth on it. And so that's a long-winded answer to say I don't really know the answer to the question.
Thank you so much. Next, we have Maria, who's asking, what is the leading theory on that mass mortality event?
Yeah, so the colleagues at Florida Gulf Coast University and the University of South Alabama have isolated a dinoflagellate, so an algae that seems to be the responsible culprit. Now, algae are when we hear about red tide and harmful algal blooms, those are harmful algae as well. This is a different one. It's more closely related to the algae that causes toxic fish poisoning, like Ciguatera, but it's not that one either. This algae apparently is usually associated with algae, larger macroalgae, as well as seagrass, and it's attached to them for some, and it's usually not in the water column. For some reason, we don't know the reason, last winter it was in the water column. And this algae has a really virulent neurotoxin associated with it. And so when the animals like sawfish are breathing in that algae, it attaches to their gills, and that neurotoxin has a really severe effect, causing this neurological disorder that seems to usually end up killing the animal. So we're a lot of the work we're going to do this winter is to monitor to see if we see that happen again, and to keep looking into what caused it this past winter and how we might be able to predict it in the future, as well as trying to figure out, what proportion of the population died.
Thank you. Chris asked if you've determined why that one sawfish went to Cape Canaveral area at the same time every year for those 3 years, whether it might be related to food, breeding, or some other reason.
Yeah, good question. Most likely is related to food. So it's not just that one, there are a lot of them. We have colleagues that work at Cape Canaveral that have a lot of receivers. That was their receivers that picked up our sawfish. And I think they've had something like 17 of our sawfish picked up on their receivers, usually starting in April and going through about July. And there are a lot of large predators that make that migration in the spring up the Florida coast. Most of the sharks keep on going, to the like blacktip sharks go all the way up to, at least to Virginia, sometimes much further than that. And they migrate through that area in huge numbers in the spring, as most people know. The sawfish do the same thing. And a lot of that is following the migration of potential prey, like the big mullet migration and things like that. It's also partly related to temperature. The sawfish, not all of them migrate up north. Some of them stay down in the Florida Keys, but the ones that stay down in Florida tend to move offshore. The coastal waters in Florida Keys get too hot for the sawfish in the peak of summer. And so either you swim north, like to Canaveral and beyond, to get into a little bit cooler water, or you go deeper. And so some do each of those behaviors. But yeah, I think it's mostly that migration's related to food.
Interesting. Thank you. Ms. Craig asks if you see a lot of other individuals doing similar things to the one being tracked in the Keys, moving east and west coastline up to the Cape and Tampa.
Yeah, so there's not very many of them have done that. Most of them, there seem to be contingents that either mostly stay on the Gulf side or mostly go on the Atlantic side. And the Keys is sort of a mixing zone for those groups. And that's true of a lot of different species. We see that with blacktip sharks and black-nosed sharks, where there are separate populations in the Gulf and on the Atlantic side of Florida, but they mix in the Florida Keys. But there are some that go to both, and there's a colleague that I'm working with. She's going to have a paper come out soon on bull sharks to show that most bull sharks are either on the Atlantic side or in the Gulf, but there are some that go into both. So, my granddad used to say, fish got tails, and that's what he would say whenever we'd go fishing and we weren't catching anything where we caught them the day before or whatever. It was just his way of saying that they do their own thing. That's not always predictable. Humans aren't predictable. No animals are completely predictable, and so they can swim, and they do, and it'll take us our lifetime to really figure out exactly why they do the things that they do.
Sure. Christine would like to know if there's any tagging in the Indian River Lagoon on the east coast of Florida.
So yeah, that's a good question. So the Indian River Lagoon um, is an area that is being worked on. I'm going to stop sharing my screen here so I can hopefully show you something. Um, Hopefully you guys can see me now. Um, this this rostrum I'm holding up here was actually caught in the Indian River Lagoon in 1872, so 150 years ago. And the grandson of the guy who caught it is gave this to me. And so sawfish used to be really common in the Indian River Lagoon. Well, when the population contracted, then they disappeared from Indian River Lagoon. Now, and this is one of our signs that the population is recovering. My colleagues at FWC have actually been tagging quite a few uh, off of in like the St. Lucie area and in parts of the Indian River Lagoon, and we're even starting to see some babies being born there. And so that area may once again become important sawfish habitat. So yeah, as a cumulative group, we have tagged sawfish in Indian River Lagoon. Mostly that's the my colleagues at FWC that have done that.
Thank you. Juniper School would like to know if sawfish can breathe through pumped ventilation.
Yeah, so they can't. So since they're a sting, basically a batoid, similar to a ray, they have the large spiracles on top. And so, rather, sharks and rays breathe in one of two ways or some combination, either ram ventilation, where they have to swim forward and force water over it through their mouth and across their gills, or they physically pump the water through the spiracles down to the gills. And that's the way the sawfish do it. And so that's one of the reasons why, when we think about their bycatch and their risk of them getting killed in a different fishery, things like long lines, we don't worry about too much because uh, they can just lay on the bottom, whereas a shark that's restricted on a line might die because it can't breathe very well. A sawfish can just lay there and breathe just fine. But then if they're caught in a shrimp trawl, then they're restricted and they can't breathe, and then that's when they're more likely to die. So yes, they do. They pump through their spiracles to pump water, oxygenated water across their gills.
Thank you. Several people in the chat are asking, how do you safely secure a sawfish?
Yeah, it's not for everyone to do, that's for sure. We don't encourage anyone to try to restrain a sawfish. If you catch a sawfish, cut the line as close as you can get it to the hook and let it go. Don't try to grab it. It really involves getting we have to get the rostrum secured first. We sort of lasso it, and then once we do that, we can get the tail up and put a rope on the tail. And then we'll usually, if it's a big animal, we'll also put a mid strap on it just to take some of the weight off, just to help secure the animal while we work it up. And then we work it up and we release it. And if anybody's concerned, we actually, these animals all have tags, and so when they're detected on a receiver, we know the animal obviously survived us catching and tagging it, and every single one of them that we've ever caught, we hear from again. So they all survive the ordeal of being caught and tagged.
Mike wants to know if sawfish lose teeth like sharks.
So sawfish do lose their oral teeth like sharks. The teeth in their mouth are like are very similar to stingray teeth. They're these little pebble-shaped teeth. Um, And they do rotate through those and lose them over the course of their lifespan. The teeth on the rostrum, they do not replace. And so they grow those throughout their lifetime. If one breaks off and it doesn't break off all the way down in the socket, then it will continue to grow. But if it breaks off in the socket, then it will never grow again. And we see them frequently, the older ones, where you can see the scar in the healed area where there used to be a tooth, but there's no longer a tooth. So they do not replace the rostral teeth.
Thank you. We have time for just a few more questions. Our next one is going to be, if the rostrum is fragile, is wondering if the rostrum is fragile, how exactly is it used by the sawfish?
So the rostrum itself isn't really fragile. It's pretty tough and it's flexible. Now, that doesn't mean it can't break. We do see sawfish with broken rostra. We've caught them with ones that are broken. If they get hung in a crab trap or a dock line or something like that, then the rostrum can be broken. But but it does. But it is pretty tough. In fact, you can tell that by the holes that they put in the gel coat of our boat whenever they if they hit the boat, which we try to avoid having them do. But if they hit the side of the boat, it will leave a hole in the boat. So it's a tough structure, that rostrum.
Fascinating. Our next question is, when is the sawfish mating season? Is it in April, May-ish?
It's spring. It's probably as early, it can be as early as February or March, but yeah, March and April seem to be the primary time that they're mating. We didn't really have a good handle on it for a while. We know when they give birth, but that's one of the things that our lab has been looking at, and we've caught males and females with mating scars. Um, in April primarily. And so that seems to be when the mating season is. In fact, one of the uh, one of the times we caught a male and two females together with mating scars 7 years ago in 2017 in the Everglades. We've been tracking those three animals for 7 years, and that male was one of the ones that died in the mortality event this past spring, unfortunately.
Thank you. Kristen asks if work has been conducted to genetically analyze connectivity between Florida population and the smaller southern populations like Cuba and the Bahamas.
Yeah, I think I saw that one come through. I think that might have been Kristen Palmrose, who used to do some of this work with me years ago. Um, so there's been genetic analysis that shows that the Bahamas and the U.S. is different. We have a whole bunch of samples from Cuba that we haven't been able to import yet to analyze, but that's what we hope to do next. But both the ecology and the genetics suggest that at least the Florida and the U.S. population are separate.
Kent asks what the depth habitat of sawfish is, and are they deep divers?
So we put satellite tags on archival satellite tags on a bunch of sawfish early on in this study, and the deepest we had any of them go was just over 100 meters, so about 330 feet or so. Most don't go that deep. The edge of the shelf at about 200 feet deep is as deep as they go. And then they spend a lot of time in very shallow water too. When we catch them in Florida Bay, they're usually in 6 to 10 feet of water. So they go from very shallow down to maybe 200 feet usually.
Thank you. We're going to wrap up with just one more question. We have some students who are wondering when you knew you wanted to study sawfish, and do you have any advice for those who might be interested in pursuing similar work in the future?
Yeah, so I grew up on the Gulf Coast. This is back home for me, and so I caught a little Atlantic sharp-nosed shark when I was a kid. That led me down the road of wanting to study sharks and their relatives. It wasn't until I came back to Florida after being in Virginia and Hawaii for a number of years, I came back to Florida that I kind of fell in my lap that I had the chance to start a project working on sawfish, which has always been one of those species that I would love to work on because they're so cool and so crazy. And so my suggestion is, if you want to work on sharks or sawfish or anything else, is to take every science course you can take every math course you can take every statistics course you can take, and then find an undergraduate program for college that has a solid biology program. It doesn't have to be marine biology, just a solid biology or zoology program, or environmental science, and then move on and try to get, while you're in undergrad, try to get as much experience as you can working in various with various researchers at your university. That's what's going to set you up for it. I went to the University of Miami for my undergraduate, and the reason I did is because Dr. Sonny Gruber, who was at the time the most famous shark biologist in the world, that's where he was, and that's why I went to undergraduate there.
Well, thank you so much. We really enjoyed speaking with you and learning from you and appreciate you joining us today. I'm going to go ahead and turn things over to Stephanie to begin to wrap up today's presentation.
Thanks, Brian, and thank you everyone for joining us here today. Dr. Grubbs, thank you very much for taking time to share your fascinating work on sawfish and sharks and more with us. If you'd like to take a look at the K-12 extension activities and resources related to today's topic, they're going to be made available along with a recording of today's talk on the UF Earth Systems YouTube channel. Also, as you can see here, for more information on the Scientist in Every Florida School program or the ANGARI Foundation, you can visit our web pages. You can follow us on social media as well. Finally, we have one more exciting event for our Fall 2024 Ocean Expert Exchange live webinar series. On December 5th, we'll be joined by explorer, educator, and science communicator Michelle Hoffman-Trotter. And Michelle will be sharing her journey to the polar seas and beyond while discussing the variety of career opportunities that exist for working at sea. So we hope you have a wonderful rest of your day and that you can join us in our future Ocean Expert Exchange. Thanks, everyone. Goodbye.